Hello everyone and welcome back for more biotechnology. In this video we are going to talk about some other things that you can do with cells, particularly bacterial cells when we talk about fermentation. And then the major focus of this fairly short video is going to be on cell growth kinetics, things to consider as you go throughout the biomanufacturing and of course the scale-up process that we talked about in the previous video. So fermentation, something that I'm sure you've heard of before, refers to a cellular process by which cells can make all sorts of useful things for us. Things like alcohol, dairy products, and other sorts of things as well. So what fermentation is a energy-making processes that cells have to go through when they are without oxygen. So this is what is sometimes also called anaerobic respiration. So the deal with this is that usually most cells will prefer to have oxygen around. Now, there of course are exceptions to that, like obligate anaerobic organisms, but in general, most organisms, most cells do like to have oxygen around because if they have oxygen around, they generally are able to make a lot more energy and get a lot more bang for their buck. So fermentation is a way to still make energy even though there's no oxygen around. You're not making a whole lot of energy, but you still get by, at least in the short term. Now, the truly interesting thing about fermentation that we as biotechnologists can take advantage of is that cells tend to make very useful things when they're undergoing fermentation. So one type of fermentation, which is called alcoholic fermentation, will produce both carbon dioxide and, more importantly, ethanol, which you know better as grain alcohol, so the basis for all of our liquors and things of that nature. So typically yeast and certain types of bacteria will perform this type of fermentation. So if you know anything about the alcohol making process or beer brewing or anything of that nature, you know that you need some type of bacteria or yeast organism in order to ferment and to actually create the alcohol that is going to form the basis for the spirituous beverage. The other major type of fermentation is called lactic acid fermentation. Now, our cells, being mammals, and then other types of bacteria will also perform this type. Same sort of thing, it's just instead of producing ethanol, you are instead producing lactic acid. So, we've said all along throughout this course that biotechnology is all about essentially tricking cells into doing what you want them to do. So, if you are in the business of making alcohol or making dairy products that are based on lactic acid and things like that, then you see an opportunity here, right? So you want to know, how can I get cells to make alcohol all the time? How can I get them to make lactic acid all the time? So it's all about understanding the biology and then manipulating the circumstances in which those cells are living so that they do what you want them to do. So you can actually force cells to undergo fermentation if you grow them under anaerobic conditions, meaning that they do not have access to oxygen. Okay, so now at this point, let's back up and talk about something that is germane to a lot of what we talked about in the scale-up process in the previous video. So whether you are doing a fermentation process or whether you're making a whole bunch of protein and you're having to scale things up at every step, it's very important to understand the kinetics of cell growth. Cells don't just grow willy-nilly, and they don't just grow at the same rate no matter what the conditions are. There, of course, are certain conditions that promote cell growth, and there are certain conditions that inhibit cell growth, and we need to understand those well if we are manufacturing something and we want that process to go as quickly and efficiently as possible. So typically, if you are culturing cells, you first seed them in what is called a starter culture. So at this point, you have a relatively low volume of culture and a pretty low density of cells. So you don't have a whole lot of cells, and they have some room to kind of spread out and grow, right? So at this point, the growth of the cells is going to be pretty slow. The density is pretty low, the volume is pretty low. So these cells are in what we call the lag phase, in which it's going to take some time for them to really coalesce into a place where they can really start dividing and growing. Now, to understand what we do about cell division, regardless of what kind of cell you're dealing with or what type of cell division you're dealing with, cell division involves the doubling of cells every time you have a division event. So it's useful to think about, in terms of time, it's useful to think about what we call the doubling time of a particular type of cell. So in other words, if you've got 500,000 cells in your starter culture, then if you know the doubling time of that particular cell type, then after that period of time, you should then have 1 million cells. And then after another period of time, you should have 2 million, and then 4 million, and then 8 million, 16 million, and so on. So if we want a particular example for something like E. coli, their doubling time in the laboratory is about 20 minutes. That sounds totally crazy, but it's true. About every 20 minutes, there should be twice as many bacterial cells as there were 20 minutes ago. Now, if we're talking about the doubling time, we're talking about what is called exponential cell growth. So for practical manufacturing purposes, this is the phase that we want our cells to be in most of the time. We don't want them to be in the lag phase, and we don't want them to be in the stationary phase that we're going to talk about later on. So as long as cells are in exponential growth, they are doubling every 20 minutes or whatever it is for whichever cell type you're going to be growing. So that's going to be as efficiently and as quickly as the cells are growing, and as the cells grow, of course, they are going to continue to make our protein of interest, right? So if you let cells go through exponential growth for too long, the density of the cells is going to get greater and greater and greater and greater until the cell density will eventually become so great that the cells are basically going to start competing with each other for resources like nutrients and space and things like that. So once those nutrients run out, the cells are not going to be growing anymore. You can't grow if you don't have any nutrients. So at this point, the cells are in what we call the stationary phase, in which the cells are just kind of hanging out. They have drastic changes in their metabolic activities. They probably aren't going to be making a whole lot of protein at this point. So once you get close to this point, it's time to scale up. So at this point, if we're growing our cells from our starter culture and now we're in, say, a 100 milliliter culture, and we get our cell density too high, it's time to scale up. So we could pour that 100 milliliter culture into say 1,000 milliliters of new LB medium, then the cell density per volume of media goes way back down, and we can get the cells to go back into exponential growth. And then we can grow those cells and let them get close to stationary phase again, and then we can scale up into an even larger volume again. So this discussion that we're having here on cell growth kinetics should give you a good frame of reference for understanding why the scale-up process is necessary and why you have to do that several times. Because if your ultimate goal is to get these cells to make a particular type of protein, you don't really want to run into the brick wall that is the stationary phase. You want these cells to continue dividing and continue making protein. Okay, and then the last thing that we'll talk about in this video is what are called good manufacturing practices, and then also we'll talk a little bit about how we go about retrieving our protein product, if that's indeed what we are making. So we're not going to go into too much detail. We can talk a lot more in Chapter 9 about how you go about purifying your protein with things like column chromatography, so you should look forward to those discussions. But the one thing that we need to understand, at least for now, is that in most cases, the protein, once you decide the cells have made enough protein, in all likelihood, that protein is still going to be inside the cell. Now, that's not always the case, so some cells may secrete their protein into the extracellular fluid. So if that's the case, you don't actually need to lyse open your cells. You don't need to blow up the whole cell to get all the goodies inside. If the protein is secreted, then all you have to do is spin down the cells and then collect the media, and your protein should be in the media. But If your protein is still inside the cell, which in all likelihood it probably is, you're going to need to undergo cell lysis and blow up those cells to release all the proteins and DNA and all that other stuff into the supernatant. And we've talked before about what sorts of things you would have to do to separate all the gunk that you don't want, like genomic DNA and membranes, away from the soluble goodies that you do want. And like I said, Chapter 9 is going to discuss different chromatography methods of purifying proteins based on a number of different physical properties, like their molecular weight, their amino acid makeup, their electrical charge, how they interact with other proteins. So those are going to be good, fun discussions, and it'll be a nice extension upon what we're talking about here, because it's the next logical step. After you've actually gone through the biomanufacturing process, now it's time to actually go about retrieving what we were trying to make all along. As far as storage goes, depending on what you're making, regardless of whether it's a protein, a nucleic acid, or something else, your product may either be stored and sold as a liquid solution. So a lot of times if you're making a protein, you may sell it as just a very highly concentrated solution of protein in a particular buffer. You can lyophilize your product, which is basically just a freeze-drying process. So if you sell a lyophilized product, then you're basically just selling someone a tube that has some powder in the bottom of it, and it's going to be up to them to resuspend that powder in buffer of their choosing. Or you can even sell your product in a pump or a patch, like what you would do with something like insulin or nicotine. So one thing to be aware of, if your product is designed for human therapeutic purposes, then the production and manufacturing and isolation all has to undergo and follow what are called current good manufacturing practices. And those, just like the clinical trial processes that we talked about earlier in the semester, are overseen by the Food and Drug Administration. Alright, so that is going to do it for this video. We've got one more video to go in this chapter, and then we will be all wrapped up, and we can move on to Chapter 9, in which we will start talking about purifying proteins with chromatography. So thanks a bunch for your attention, and I will see you next time. Bye-bye.